actually myself working in melanoma, and I have a really quite of exciting question. Since I'm working actually in mice, it's really difficult to unravel what kind of immune cells do you have inside your tumor microenvironment. This is in mice actually. And since there is, like, you can find in the tumor microenvironment, it's very heterogeneous. And I've been actually struggling to know what is the answer to a really interesting question. Is we can find a different phenotype of immune cells, and then we cannot be like, for example, you have the classical subtypes of cells, and then you find another immune cell that looks like the classical T cells that are basically defined, and then you don't know if it's really a cell type or a cell state. And in this regard, actually, my question is, can a heterogeneous immune environment be deleterious for the effective immune response against cancer progression? I mean, making these cells interact with the tumor cells, is these cells are really a cell type or just a cell state that would, I mean, like, it's a cell state, and then the cells will differentiate to another cell type, which would be like kind of new cell type of the classical cell type that is known before?
Yeah, these are really great questions, Dounia, that you are asking, and it's very tough questions to answer. You're talking about the cell plasticity and whether it's a fixed state or cells are undergoing different differentiation stages. They're very tough questions to answer. I think we have still many questions that we should try to answer within the tumor microenvironment. As you know, there are plenty of cell types. Of course, there are immune cells, T cells, NK cells, NDSC cells, NKT cells, so many immune cells, but also non-immune cells, which also can play a major role. So I think we are just at the stage where we need to decipher all cell types within the tumor microenvironment, at least trying to understand the cells that are, pro-tumoral, and then the cells that are anti-tumoral, or the cells that are pro-immune cells that can eliminate the tumors, and cells that are deleterious. I think we're just at the beginning of understanding all these questions, and I think this is where our lab actually try to, to bring its piece of discoveries, is by trying to understand all these different types of cells. And another point that you actually I wanted to bring here is, it's really important to work on mice, but we need to translate that in humans as well. So each time that we learn things from mice, we need to translate that and see whether it really translates, well in humans, to make sure that there is an analogy between what happens in mice and in humans. Of course, we need to work on mice as well, because many questions we would not be able to answer many questions in human. We need mice to do that as well, but we need each time to confirm that and to corroborate that indeed it works the same way in humans as well.